Dear students, my name is Günel Əyyubova. I am senior lecturer of Department of Histology, Embryology and Cytology of Azerbaijan Medical University. And today I'm going to talk you about the cardiovascular system, its development, general morphofunctional plan, relations between hemodynamic conditions and structure of the vascular wall, heart, arteries, microcirculation network, capillaries, venous network, anastomosis, neurohumoral regulation of the cardiovascular system functioning, age changes, and regeneration abilities. As we know, the circulatory system of the human body, it comprises two systems. It is cardiovascular system and the lymphatic system. Cardiovascular system, it provides bidirectional flow of fluid, which is blood, from heart and to the heart. And lymphatic vascular system, it provides unidirectional flow of fluid, which is the lymph, from tissues towards the cardiovascular system, and by that way lymphatic system returns tissue fluid into the veins, into the venous system. Cardiovascular system, its center is the heart, which pumps blood into the closed system of the blood vessels, and by that way it provides bidirectional flow. Arteries, these are vessels of, which carry blood from heart, and veins carry blood to heart. That's why the arteries, these are vessels of high pressure, and that's why their wall is will correspond to their functions. Their wall is thicker than the walls of the veins. Also, the arteries, their wall is rich in elastic components, which are needed to withstand high pressures. The conducting vessels, the arteries, when they go to the organs, they undergo branching, repeating branching pattern. Finally, they lead to the smallest arteries, which are arterioles. Arterioles in turn branch, and finally they lead to metarterioles, and finally to capillaries. The capillaries are smallest vessels of the vessel system, which have thinner walls to provide exchange of substances between the blood circulation and the surrounding tissues. Capillaries, they join each other to form venules. Venules will increase in diameter with each convergence, and we will have small, medium-sized, and large veins, which will converge. Finally, in the body, we will have two larger veins, which are superior and inferior vena cava, that carry blood, return blood to the heart. Because the veins, these are vessels which carry blood with low pressure, their wall is thinner, but their lumen is larger. That's why they are vessels the vessels of that may accumulate stored blood. So more than 70% of blood of the body is stored within the veins. Then we also have arteriovenous anastomosis that will carry blood from the arteriolar end to the venous end, bypass capillary network. They also have their very important functions. Here we are looking at the similarities in the vessel wall, the layers in the vessel wall of arteries and veins, but at the same time they have very dissimilarities. Here we are looking at the arteriovenular anastomosis and the capillary network here. In the wall of the capillaries, we will have additional cells. These are pericytes that surround capillaries, and the amount will increase in the postcapillary venules. Okay, now we are looking at the cross-section of the artery and the vein. As we see here, the wall of the artery is thicker than the wall of the vein. Also, we have to take in mind that the outer coat of the vessels is fused with the surrounding connective tissue, which will restrain the vessel wall from the and will prevent damage and over dilatation or collapsing of the vessels. The innermost coat of the vessel wall, that is the endothelial lining. Endothelial cells, they form one layer of squamous flattened polygonal cells that align their long axis will be along the direction of the blood. Endothelial cells, they join each other via tight junctions. Also, there are desmosomes and gap junctions. Inside of the endothelial cells, we will have pinocytotic vesicles, which will provide exchange of substances between the blood and the surrounding tissues. The membrane of the endothelial cells will have a lot of receptors for different substances because endothelial cells are in contact with the blood which flows. They are main cells that interchange with blood. It is interplaced between the blood and the tissues. That's why they have a lot of carrier proteins, receptor molecules, and also adhesive molecules. If we are talking about the functions of the endothelial cells, we have to mention different its functions. First of all, in the normal state, endothelial cells produce anticoagulant factors, that is antithrombin third and thrombomodulins, prostacyclins, these substances needed for prevention of the clot formation at the normal state and the normal conditions. But if the vessel wall will be damaged by different sources, the endothelial cells produce von Willebrand factor. Normally it is stored inside of the vesicles in cytoplasm of endothelial cells, but during damage of the wall of the vessels, they released into the blood circulation, cause coagulation, clot formation, aggregation of the platelets, and other thromboxanes and other coagulation factors also are released from the endothelial cells if it is needed. Then angiogenic factors. Endothelial cells produce vasoactive endothelial growth factor, which is very important factor for the formation of the new blood vessels. And platelet-derived growth factors, fibroblast growth factors, endothelial growth factors also needed for this purpose. Endothelial cells also produce substances to increase permeability of the vessel wall, that are nitric oxide, prostaglandins I2, reactive oxygen species. If we are talking about these components, we have to mention vasodilatators and vasoconstrictors. Vasoconstrictors will lead to decrease of the lumen of the vessel, to decrease amount of blood that circulates, that comes to the corresponding organ. These are angiotensin second. It is produced from the angiotensin first under the influence of the angiotensin converting enzyme that is present in the endothelial cells of the mainly lung capillaries. And then endothelin 1, it is also potent vasoconstrictor substance, thromboxanes and other components which will lead to diminishing the lumen of the diameter of the lumen of the blood vessels, and vasodilatators like nitric oxide. Under the influence of different factors, endothelial cells produce NO that diffuse through the endothelial cells to the surrounding smooth muscle cells and will activate smooth muscle cells to relax. So the smooth muscle cells will relax and it will lead to dilation of the blood capillary, and by that way amount of blood which comes to the organ will increase. Then prostaglandins I2 and other vasodilators also produced in a certain amount. Finally, we are coming to the inflammatory activity of the endothelial cells. So we have to mention here that here produce very important inflammatory mediators like leukotrienes, chemoattractants that will attract white blood cells to the site of inflammation. And then we have here a lot of cell adhesion molecules like selectins, integrins, immunoglobulins, interleukins. They are also needed to attract the circulating leukocytes like neutrophils, eosinophils, and other white blood cells, which will then come to the vessel wall. Then they will roll there and they will adhere to the vessel wall, and when they receive signal from the tissue, they will leave bloodstream, enter the tissue to perform their functions there. And the process of migration of the white blood cells between endothelial cells, that process is called diapedesis. Diapedesis, here you can see. So the inflammatory response, we will see capture and rolling of white blood cells, adhesion there via intercellular adhesion molecules, then their diapedesis. And another main function, regulation of the blood homeostasis here, then barrier, endocrine, paracrine, autocrine functions by production of different factors just I mentioned. And then vessel remodeling, formation of the new blood vessels, it's vasculogenesis, angiogenesis, arteriogenesis, so the formation of the new blood vessels. So they are the main functions of endothelial cells. Besides, we have to mention that these cells produce substances for deactivation of the inflammatory mediators like bradykinins, histamines, serotonins, noradrenalines. They are deactivated by the endothelial cells when it is needed, of course. Finally, endothelial cells produce lipids, that is the enzyme that will break triglycerides, lipoproteins, to the fatty acids and the glycerol to be stored within the fat tissue. Vessel wall. Here we are looking at the general principle of structural organization of the vessel wall. As we know, the wall of arteries and veins is composed of 3 layers. These are 3 tunics: tunica intima, tunica media, and tunica adventitia. The innermost layer of the tunics, that is tunica intima, and it is composed of a simple squamous epithelium or endothelium that lines the lumen of the vessel. And that is, that endothelium, it rests on the basal lamina. Pay attention, that is basal lamina of endothelium, which is blue colored here. Then under the basal lamina, we have subendothelial connective tissue. Subendothelial connective tissue here, it is that is like red colored thing here. That is subendothelial connective tissue. And it is surrounded by internal elastic lamina. That is perforated elastic membrane composed of mostly of elastin. The thickest of the 3 coats, especially in arteries, it is the tunica media that is composed of multiple layers of smooth muscle cells arranged in helical configuration, as we can see here. The extracellular matrix of the tunica media contains elastic fibers, then types 1st and 3rd collagen fibers, and ground substance. We have to remember that all of these components are produced by the smooth muscle cells, which are present here in the tunica media. The outermost layer of the media, and at least in large muscular arteries, houses slender elastic fibers composing the external elastic lamina. Here, the blue perforated thing, that is the lamina, which is external elastic lamina. So we have internal and we have external elastic laminas. The outermost coat, the tunica adventitia, is a fibroelastic connective tissue that affixes blood vessels to the surrounding structures. In large blood vessels, the nutrients and oxygen that is present in the in the lumen of the blood vessels, it will reach the tunica intima, then innermost layers of the media, if it is the case of arteries, because inside of the arteries flows arterial blood. That's why via diffusion, these necessary substances can reach the intima and innermost layers of the media. But the outermost coats cannot receive those nutrients. That's why, for that reason, in the wall of the vessels, we will find vasa vasorum. These are vessels of vessels which penetrate here through the adventitia and branch here. They will supply tunica adventitia and outermost layers of the media. But in case of veins, all of the tunics will include vasa vasorum, because in veins flows venous blood. That's why it is, and that's why in veins the vessel wall cannot receive oxygen via diffusion. That's why all of the layers will contain vasa vasorum. The nerve supply of blood vessels also enters the tunica adventitia, and pay attention here. These are the nerves which enter here. The vasomotor nerves release the neurotransmitter norepinephrine, which diffuses to the smooth muscle cells of the tunica media. These are sympathetic vasomotor fibers that cause the smooth muscle cells to contract, and the wave of contraction is spread via gap junctions between neighboring smooth muscle cells, and that will lead to vasoconstriction. So sympathetic fibers will lead mainly to vasoconstriction. Then now let's come to individual blood vessel types. First of all, the arteries. Arteries are the large muscular blood vessels that gradually decrease in diameter as they carry blood away from the heart and deliver it into capillary beds. Although the definitions are not clear-cut, there are three categories of arteries determined by the diameter, wall thickness, and the other histological features. They are elastic arteries are the largest, then arterioles are the smallest, and muscular or distributing arteries range in size between the other two types. And so now we will talk about elastic arteries, as you can see here. To elastic arteries belong aorta and the branches originating from the aortic arch. These are common carotid artery and subclavian arteries. Then the common iliac arteries and the pulmonary trunk also belong to elastic arteries. If we will look at the wall of these elastic arteries, the tunica intima of the elastic arteries is composed of the endothelium, simple squamous epithelium that is supported by a narrow layer of underlying subendothelial connective tissue containing few fibroblasts, occasional smooth muscle cells, and collagen fibers. Thin lamina of elastic fibers, the internal elastic lamina, are also present. The tunica media of the elastic arteries consists of many fenestrated lamellae of elastin, known as fenestrated membranes, that alternate with circularly oriented smooth muscle cells. Pay attention here. The yellow-colored substances here, these are elastic components. So internal elastic lamina we can see here, and the external elastic lamina also. Between smooth muscle cells here in the tunica media, we can see fenestrated membranes. We can also distinguish in the Orsean stained wall of the aorta here. All of these undulated fibers components, these are elastic components. Then we are looking at them. So the number of these fenestrated lamella, they are increased with age. And if there are approximately 40 lamella of fenestrated membranes in newborn, then 70 they are in adults. The extracellular matrix that is secreted by smooth muscle cells composed of the chondroitin, sulfate, collagen, and reticular and elastic fibers here. So an external elastic lamina, as we can see here, then the tunica adventitia of elastic arteries is relatively thin and is composed of also loose connective tissue. The vasovasorum also abundant throughout the tunica adventitia, as we can see here. Now let's come to the muscular arteries. The muscular arteries, examples of them, these are brachial, ulnar, renal arteries, femoral arteries of the body. So they are located far away from the heart. That's why they need more muscular elements in their wall to be able to convey blood. The identifying characteristic of the muscular arteries is relatively thick tunica media composed of mostly of layers of the smooth muscle cells. The tunica intima in the muscular arteries is thinner than that in the elastic arteries, but the subendothelial layer contains a few smooth muscle cells. Also, in contrast with the elastic arteries, the internal elastic lamina of the muscular arteries is prominent and displays an undulating surface, especially in the case of the slight histological section of the muscular arteries. Pay attention to internal elastic lamina, and it has undulating surface to which the endothelium also conforms. So endothelium also will conform to the shape of this internal elastic lamina. The tunica media of muscular arteries is composed of predominantly of smooth muscle cells, although these cells are considerably smaller than those located in the walls of the internal organs. The orientation of most of the smooth muscle cells is circular here. Each smooth muscle cell is enveloped, that by the external lamina, but it doesn't matter. Between cells, there are gap junctions that provide coordinated contractions within the tunica media. The external elastic lamina is identifiable in histological sections of larger muscular arteries, and so the tunica adventitia of the muscular arteries consists of the elastic fibers, collagen fibers, ground substance composed mostly of dermatan sulfate and heparan sulfate. These extracellular matrix components here are produced by fibroblasts. Here, pay attention, you can see the fibroblast cells. And finally, we are coming to smallest arteries. These are arterioles. Here we can see in the cross section, we can see arteriole, and we can compare them, the venules and then the lymphatic vessels. Arterioles are the terminal arterial vessels that regulate blood flow into capillary beds. So after the arterioles, that come capillaries, and these are the tubes. These are the vessels which control blood flow within the capillary beds. In histological section, the width of the wall of the arteriole, as we can see here, is approximately equal to the diameter of its lumen. So the thickness of its wall is approximately equal to the lumen diameter. The endothelium of the tunica intima is supported by thin subendothelial connective tissue layer, and here the thin fenestrated internal elastic lamina is present in larger arterioles, but absent in small and terminal arterioles. The tunica media may include 1 to 3 layers of smooth muscle cells. It depends on the diameter of the arteriole. The tunica adventitia is also thin, very scant, and represented by fibroelastic connective tissue. So the capillaries. Capillaries, these, as you know, these are smallest blood vessels with the thinnest walls. They are composed of simple squamous epithelium or endothelium that is fashioned into a tube. Here we are looking at the scanning electromicroscope representation of the capillary wall. Capillaries form the basal lamina that coats their abluminal surface. and pay attention to the cross section here of the capillary. So it's all composed of mainly endothelial cells that are joined at this region. We can see the intercellular junctional complexes, tight junctions mainly here, and the endothelial cells surrounded by basal lamina. But pay attention to other cell type which is also present in the capillaries that are the pericytes. The pericyte cell is contractile cell which is include actin myosin complexes. That's why they will control the lumen of the blood capillary. More amount of pericytes we can find in venules especially. And each pericyte cell is surrounded by the basal lamina, and the places where basal lamina of pericyte and basal lamina of endothelial cell come into contact with each other, they form basement membrane. So you know that by fusion of two basal laminas there is formed basement membrane. So that is a pericyte. Also pericytes, we have to know they form gap junctions with endothelial cells, and they will take information from the endothelial cells, and so they are interconnected with each other. It's very important in the regulation of the blood flow. Pericytes may also function as regenerative cells, and they will participate in regenerating, repairing damaged blood vessels. As the blood pools from the capillary bed, it is discharged into small venules. So we are coming to venules. Here you are looking at the cross section of the arteriole, venule, and lymphatic vessel. Arterioles and venules, we can compare them. Venules, they are the diameter more than in arterioles or in capillaries, and their wall is also thin, but in venules their diameter approximately from 15 to 20 micrometer in diameter. Their walls are similar to those of capillaries, with a thin endothelium surrounded by reticular fibers and pericytes. The pericytes of postcapillary venules form a network surrounding the endothelium. Pericytes are placed by smooth muscle cells in larger venules. So instead of in larger venules, instead of pericytes, we will see smooth muscle cells. Then as venule diameter increases, the smooth muscle cells also become more closely spaced and forming a continuous layer in the largest venules and small veins. And now we will talk about the classification of the capillaries. In the human body, we can distinguish three types of capillaries: continuous, fenestrated, and sinusoidal capillaries. Continuous capillaries, they have any pores and fenestrae in their walls, as you can see. Endothelial cell and the basement membrane also, they have not any pores or interruptions in their wall. They are present in muscle, nervous, connective tissues, as well as in lungs and exocrine glands. And in this case, the endothelial cells, they join to each other via tight junctions. These tight junctions, as you know, they seal intercellular space. Continuous capillaries were important in the formation of the barriers. For example, blood-brain barrier, blood-thymus barrier, and in other places of the body, and they will isolate the lumen of the vessel from the surrounding tissue. That is important in the functioning of those organs, for example, in central nervous system. Then fenestrated capillaries, they are located in endocrine glands, in pancreas, and in intestines, and also in the kidney glomeruli, where it is very important, more intense exchange of substances between the blood and the surrounding tissues. For example, if we are talking about endocrine glands, as you know, they release secrete their hormones directly to the blood circulation. That's why fenestrated capillaries will need some fenestrations or pores in the endothelial lining, and these pores, they have diameters from 60 to 80 nanometer, and they mainly covered by diaphragms. But in case of the kidney glomeruli, there are not any diaphragms, and these pores, they are situated at 50 nanometer intervals from each other. Also, they may be organized in clusters. It depends on the organ. Sinusoidal capillaries located in bone marrow, spleen, liver, lymph nodes, and they are formed into amorphous channels or sinusoids that lined by endothelial cells, and these endothelial cells have numerous large fenestrae pores, can you see, and they are not covered by any diaphragms. In some instances, the basal lamina and the endothelial wall may be discontinuous, and that facilitate a much freer exchange of materials between the blood and tissues. It's very important, for example, in hemopoietic organs like the thymus, like the bone marrow, the spleen, and lymph nodes. Here, the whole cells, blood cells, may circulate from the bloodstream into the tissues and in opposite directions. Also, it's important in the liver functioning. So these were the types of the capillaries. Now let's a little information about the anastomosis that will connect them. They will also have important function in the regulation of the blood flow into capillary bed, and that is here you are looking that in some cases between the arterioles and the venules we will have arteriovenular venous anastomosis. That is, for example, collateral channel, that is arteriovenular anastomosis. Also, the central channels. First of all, let's concentrate on the arteriovenular anastomosis, AVAs. AVAs, they bypass capillary beds. Instead, there is a direct connection between the arterial and venous sides. The connecting vessel possesses three regions: an arterial end, then venous end, and the intermediate segment. The intermediate segment has a thickened tunica media and modified smooth muscle cells in the subendothelial rear, and they have rich adrenergic and cholinergic nerve supply. You know, adrenergic is from sympathetic, cholinergic from the parasympathetic nervous system, and they controlled directly by the thermoregulatory centers in the brain. Blood flow is controlled by opening or closing these AVA shunts. When AVA shunt is closed, blood flows normally through the capillary bed, and when the shunt is open, blood bypasses the capillary bed. The blood will flow directly from the arteriolar vessel towards the venular vessel, and it will bypass capillary bed, and that is important in some cases. Although AVAs are located throughout the body, they are especially common in the skin, where they function in thermoregulation. For example, in tips of the fingers, in tips of the nose, they are located, and if there is cold outside, we know that fingertips become cold, and nose tip also. Why they become cold? Because their capillaries undergo contraction, and the blood will flow into the internal organs capillaries, and if there is hot outside, these capillaries of the tips of those in the fingers tips and in nose tip, they will open, and the blood will flow through all of the capillary bed and blood, by that way it will lead to evaporation of the heat and losing of the heat from the body. It also has very important role in the thermoregulation of the body. And then the central canal. Pay attention, the central channels are composed of the metarteriole. Pay attention, metarteriole, and its continuation, the thoroughfare channel. Pay attention here. The metarterioles, they arise from the arterioles and possess precapillary sphincters that when they open, they allow the flow of blood into the capillary bed. Blood from the capillary bed then enters the thoroughfare channel. Pay attention. From the capillary bed, then blood will flow into the thoroughfare channel, and because these channels do not have sphincters, blood can always enter them, and from there blood is delivered into small venules. So that is the microcirculatory bed, which we have to know very well, and these vessels will provide our organs, our tissues with blood supply. Now we are coming to the veins. The vein, you know that capillary beds deliver the blood to venules from which the blood drains into veins of increasing size until it enters the atria of the heart. Because veins are low pressure blood vessels, there are more veins than arteries, and their luminal diameter is greater, such that they contain approximately 70% of the total blood volume. Veins and arteries are usually side by side, but the walls of veins are flattened. Pay attention. They are flattened because their walls are thinner and less elastic, because they include less elastic elements than in the case of arteries. If we will compare these two vessels, the wall of the arteries, because it is thicker and include more elastic elements, in cross sections, arteries have more rounded appearance, but veins will have collapsed, flattened appearance. Also, here, although veins possess the same three tunics as arteries, the boundary between the tunica media and the tunica intima is relatively indeterminate. We cannot distinguish the boundary between the intima and media, and but the tunica adventitia is increased in thickness. So if in arteries the thickest layer, thickest tunic is the tunica media, then in veins the thickest in the wall of the veins, that is the tunica adventitia. Then veins are classified also into three groups. They are venules, medium and small veins, and large veins, and the large veins. And because of them thin wall and the low pressure, medium-sized veins, especially the veins of the lower extremity, possess also valves that ensure a unidirectional flow of blood. So one of distinguishing feature of veins from the arteries, that is the presence of valves in some of them, mainly in those which carry blood from the lower parts of the body. Then we are coming to the heart, that is the center of the cardiovascular system, that is the pump which will push the blood into all of the circulatory system. So the heart is the pump responsible for the circulation of blood throughout the body, and to accomplish that task, it has four chambers. The two atria which receive blood from the venous system, and the two ventricles, which propel the blood from the vessel from the heart to circulate throughout the body. The four chambers are divided into two circuits, a pulmonary circuit and systemic circuit. So here we also can distinguish the valves which present between the atria and ventricles. That is a cross section of the heart. Here you can see the mitral valve, bicuspid or valve, a tricuspid valve in section of the heart. We can see here. Now we will look at the structure of the heart wall. Actually, we have to know that the heart is highly modified blood vessel. It's like a modified blood vessel. We can look at it like this. It possesses three layers. That is the endocardium, then myocardium, that corresponds to the tunica media. By the way, endocardium, it corresponds to the tunica intima. Myocardium, it's like tunica media of the vessels, and then the epicardium, that is like tunica adventitia. The myocardium is composed of the cardiac muscle, as we know here, and so that is the heart. Enclosing the entire heart and becoming continuous with the visceral pericardium on the great vessels, entering and leaving the heart is the pericardium. So we will look at the schematic representation, and we will find out what is the pericardium and which layers it has. Actually, we have to know here that the heart is located within the cavity, and to understand it, we have to know that there are two types of pericardium. That is fibrous pericardium. In this illustration, it's number 6 indicates. Then we have the fibrous pericardium, and we have serous pericardium. Serous pericardium has two layers. It is visceral layer, number 3 here, and the parietal layer, number 5. Between these two layers of the serous pericardium, so visceral and parietal layers, located cavity. That cavity is called pericardial cavity, number 4 here, pericardial cavity. It is a cavity which includes fluid that will facilitate pumping activity of the heart, so heart can move because of this the structural arrangement. In larger view of this quadrangular region, we also can distinguish the same layers, endocardium, myocardium, and then we can distinguish here fibrous pericardium, number 12. Then the layers of serous pericardium, that is number 11, parietal, and number 9, visceral layers. Between them, pericardial cavity. Also, we have to know that visceral layer of serous pericardium we can consider as epicardium. So in this case, number 3 here will indicate it. So the epicardium, actually, that is visceral layer of the serous pericardium, which form the third tunic of the heart wall. In the section of the heart, we also can distinguish the same layers. So the endocardium here, then myocardium and epicardium. Then we are looking at the valve, for example, mitral valve, and valves. These are extensions of the tunica of the endocardium, and they also will have this. They will covered by endothelium, and inside of each valve we can distinguish just connective tissue elements. They are avascular, by the way. Then all of the valves arise from the fibroskeleton of the heart. That is, you have to know that heart includes its own skeleton, which composed of the dense connective tissue that forms in the wall of the heart. Fibrous skeleton, that are the rings, will be in the basis, at the basis of the valves of the heart and in the base of the large vessels that arise from the heart. Then two triangles, right and left triangles. Then finally the fibrous part of the septum membranaceum between the ventricles, right and left ventricles. So that will be fibroskeleton, and from where from where will arise also the muscles of the heart. They will provide independent attachment of the muscles of the atria and the ventricles. Then let's look at the structure of each of these tunics. First of all, endocardium. Endocardium is the innermost layer which covers the heart from internal its side. It is composed of, actually it is lines the lumen of the heart, and because it is continuation of the tunica intima of the blood vessels, it is composed of squamous epithelium, which is endothelium, and which lies a fibroelastic connective tissue with a scattered collection of the fibroblasts. A deeper layer of dense connective tissue is richly supplied with elastic fibers and intermingled with the smooth muscle cells. The deepest layer, the subendocardial layer, separates the endocardium from the myocardium, and it is composed of loose connective tissue with blood vessels, nerve fibers, and the Purkinje fibers. So these are the layers of the endocardium. And just I mentioned that in subendocardial layer we will have also Purkinje fibers. So what are the Purkinje fibers? We will look on next slide. The endocardium we can see here, endothelium, connective tissue, then the myocardium, and between these two, endocardium and myocardium, we can see subendocardium or subendocardial layer, which includes Purkinje fibers. In this section, we also can see the same things, endothelium, connective tissue, and let's look at the Purkinje fibers. Purkinje fibers, these are the cells which belong to the conducting system of the heart. They are actually modified cardiac muscle cells which form then like nervous apparatus of the heart. These are modified cardiac muscle cells which will form the conducting system. Actually, these cells are larger, they are clumped, they are rounded shape they have. They are larger than conventional cardiac muscle cells which form myocardium, and they have nuclei were prominent. Some of them may be binucleated, so they will branch in this layer, in subendocardial layer. If we will look at the conducting system of the heart that is formed by these cells, we will see this appearance. The conducting system of the heart, so it is formed by Purkinje fibers, and it includes synovial node, then atrioventricular node, then atrioventricular bundle or his bundle, then Purkinje fibers. This will branch into right and left branches. Then pay attention, there are bundles that will branch into, can you see these branches that will enter into the subendocardial layer, and they will carry impulses. The impulses actually they arise in sinoatrial node. We will concentrate here. The sinoatrial node or pacemaker, because here located pacemaker cells, they generate impulses for contraction, and they will generate impulses at the rate 70 to 80 beats per minute, and that node is located at the junction of the superior vena cava and the right atrium. These specialized nodal cardiac muscle cells, so they can spontaneously depolarize 70 to 80 times per minute, creating an impulse that spreads over the atrial chamber walls by internodal pathways to the atrioventricular node that is located in the septal wall just above the tricuspid valve. Modified cardiac muscle cells of the atrioventricular node regulated by impulses arriving from the sinoatrial node, and they will transmit signals to the myocardium of the atria via atrioventricular bundle or bundle of His. Fibers from atrioventricular bundle pass down the interventricular septum to conduct the impulse to the cardiac muscle. So they will produce a rhythmic contraction. This atrioventricular bundle travels in the subendocardial connective tissue as large modified cardiac muscle cells, just we told about, that are the Purkinje fibers that transmit impulses to the cardiac muscle cells located at the apex of the heart. And the Purkinje fibers are have to be not confused with the Purkinje cells in the cerebellar cortex, actually, take in mind. So stimulation of the we have to know that in the regulation of the heart beating also participate sympathetic and parasympathetic nervous systems. Actually, the impulses will be generated within the heart itself because it has pacemaker cells, but the autonomic nervous system will modulate the functioning of the conducting system of the heart. When increased sympathetic activity, it will simulate production of the generation of the impulses in the heart. So it will lead to increase of the heart rate, the power of contractions, but activation of the parasympathetic nervous system will slow down rhythmic activity of the heart, and it will lead to decrease of the rate of the contractions and the power of heart contractions. Now let's come to the histological structure of the myocardium. The myocardium is the middle and the thickest layer of the heart wall is composed of the cardiac muscle cells organized in spirals surrounding each of the 4 chambers of the heart. Cardiac muscle cells have various functions. Some of them join the myocardium to the fibroskeleton of the heart, and I mentioned that some of them they are contractile, and the cardiac muscle tissue we studied in previous semester, and you know that that is striated muscle. It's type of striated muscle tissue. Here the individual cardiac muscle cells or cardiomyocytes, they join each other via intercalated discs that you can see here, these regions of intercalated discs. Then between they will form functional syncytia. They join each other and form functional syncytia. They form like networks. In these intercalated discs, we have some types of intercellular junctions, including gap junctions, by which the signals for contraction, they will spread from one cell to the neighbor cell, and all of the muscles of the myocardium have to contract at the same time. That's important for the proper functioning of the heart as a pump. Between these cardiac muscle cells, we can distinguish capillaries that include the blood cells, that is the loose connective tissue that carry blood, capillaries. And also finally, we just mentioned that we have also modified cardiac muscle cells which form conducting system of the heart. Then we have cardiac muscle cells that synthesize and secrete hormones such as atrial natriuretic polypeptide or cardionatrine or atriopeptin. These hormones function in maintaining fluid and electrolyte balance and reducing blood pressure. they lead to release of the sodium ions from the body. They target the targets of those hormones. These are kidney tubules. Then we are coming to epicardium. The epicardium, it is the outermost coat of the heart, and it is actually it is visceral pericardium, just I told you, that consists of it consists of the mesothelium, simple squamous mesothelium that is supported by a layer of connective tissue containing, here you can see CT, that is connective tissue, and inside of the epicardium, pay attention, that is myocardium, and that is epicardium. Okay, so simple mesothelium, then connective tissue. Inside of the connective tissue, we can find nerves, then fat cells, adipose cells. These are adipose cells, and so that is characteristic feature of the epicardium. Also, epicardium contain coronary vessels that branch here. So nerves, ganglia, they will present in epicardium. And finally, we are coming to the specialized sensory components which are present in the vessel wall. And so in the wall of the large vessels, large arteries, we have sensory components, vascular afferents that serve as receptors to sense composition of the blood, its chemical composition, and also pressure inside of the vessels, to carry the information to the brain, to the vasomotor centers, to the respiratory centers, and by that way the brain will control the blood pressure, heart rate, and the respiration. These sensory components are carotid bodies. Carotid body, which is located at the bifurcation of the common carotid artery. Then we have also aortic bodies, which present in the arch of aorta. Here present receptors, which are chemoreceptors that will sense chemical composition of the blood, sense oxygen, level of oxygen, carbon dioxide, and pH of the blood. Then we have here the nerve endings, nerve fibers, which belong to the glossopharyngeal nerve, and these nerves will carry information, sensory information, to the brain. So that is the carotid body. We have also carotid sinus. The carotid sinus, it's located in the wall of the internal carotid artery, just above the bifurcation of the common carotid artery. And the sinus, it contains nerve fibers that sense the pressure of the blood. So here are present baroreceptors. And here, visceral sensory carotid sinus, it will be branch of the glossopharyngeal nerve and also vagus nerve. From in this region, we have to mention that tunica, under the endothelial lining of the blood vessels, present polygonal modified smooth muscle cells, and also here in this region the tunica media is thin, but tunica adventitia thick, and here present nerve endings, nerve fibers, to sense the blood pressure. That is the aortic bodies, and here we have carotid bodies, and we have to talk about if we are talking about the structure and composition of the carotid bodies, we have to mention that it has some specialized structure. There are present two kinds of cells, which called glomus cells, these are, and also we have sheet cells, sheeted cells which will cover the glomus cells. So glomus cells here we are looking, and sheath cells or sustentacular cells. This is second type of cells that surround glomus cells. They are like glial cells. But glomus cells, they contain granules inside of which is accumulated catecholamines, for example, norepinephrine. And here we are looking also at afferent fibers, also glossopharyngeal nerve, that come and form synaptic connections with these cells. Here also present the blood capillaries. So in response to the blood pressure inside of these capillaries, the glomus cells produce catecholamines that is released into the nerve endings and will excite these nerve endings. So the nerve fibers will carry nerve impulse to the brain to inform the brain about the blood pressure. So that is all for the cardiovascular system. Thank you for attention.